Hello everyone, and welcome to today's live broadcast, Mammalian in Vitro Translation, Cell-Free System for Rapid Protein Expression and Production of Therapeutic Proteins. I am Soon Pham of LabRoots, and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar sponsored by Gibco, part of Thermo Fisher Scientific. For more information, please visit www.thermofisher.com/proteinexpression. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the green Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window or submit your problem through the green Q&A button lower left. Now let's get right to today's presenter. We're proud to welcome Dr. Penny Jensen. Dr. Jensen is a research scientist at Thermo Fisher in Cell-Free Protein Expression Group. Her exciting research includes the use of one-step CHO high-yield IVT system to express protein and biologics on demand in collaboration with DARPA, the Defense Advanced Research Projects Agency. Previously, Dr. Jensen was a senior scientist at Sigma-Aldrich, where she developed tools for the shRNA library. She completed 2 postdoctoral positions at Washington University in St. Louis studying Parkinson's disease and diabetes. I will now turn it over to Dr. Jensen for her presentation.
Thank you and welcome. I'm very excited to be here today to have the opportunity to tell you about our fabulous protein expression products. Our products utilize mammalian vitro translation, also known as cell-free, for rapid protein expression and is used for the production of therapeutic proteins. Proteomics is the study of proteins, investigating their protein structure, function, and regulation. One invention that has greatly enhanced our ability to study proteins is the use of recombinant proteins. And recombinant proteins have quickly become central to proteomics research. They have been used for enzymatic assays, activity assays, the investigation of post-translational modifications, use for assay standards, and antibody production. Options to express recombinant proteins vary. You could use in vivo expression. You can express in bacterial systems where E. coli is the most common, yeast with the use of Pichia pastorius, insect cells that commonly use SF9 cells, or mammalian cell culture that use CHO or HEK293 for their workhorses. Alternatively, one can express the recombinant proteins through in vitro protein expression. What is in vitro protein expression? The cell is an amazing machine. It produces many different types of proteins all the time. And how does it do this? It does this by reading the DNA blueprint in the nucleus and transferring that through transcription into RNA through the use of RNA polymerases, nucleotides, and energy. RNA is then moved out of the nucleus and translated into proteins through the use of translation factors, tRNAs, amino acids, ribosomes, and also energy. What we have done is we have harnessed this workflow by removing the nucleus and taking the translational machinery and putting it into a tube in a cell extract, allowing us to express proteins in a tube. The advantages of using in vitro translation is number 1 is speed. You can produce proteins in a couple of hours compared to the days that it will take for you to express proteins in mammalian cell culture. Because you don't have to keep a cell happy, you have the ability to express toxic proteins and the ability to incorporate non-natural amino acids into the proteins. The speed enables rapid protein engineering and mutational analysis. Recombinant proteins expressed in IVT have been used in NMR, mass spec, and crystallography studies. Just like expression in vivo, there are different types of IVT systems available. You can express your recombinant protein in bacteria or plants where you will get a high yield of protein where you will suffer poor modifications and insolubility issues. You can express in insect systems where you will take a hit in yield, but you will get some glycoforms. rabbit suffers from low yield. We have developed two systems that give you what is needed to get good protein. One is CHO and one is human. And these give you what is needed, high yield and the addition of post-translational modifications. What types of systems do we have available? We entitled our IDT systems One Step to convey the simplicity of our system in harnessing the power to make proteins into one quick and easy step. First, we have our one-step coupled IDT kit. And this is a human-based system for the expression of small-scale protein to do experiments such as electromobility shift assays, co-IPs, I routinely quickly express proteins to be used for Western blots, and mass spec. For these systems, we have two different types. We have a human high-yield IDT kit and a CHO high-yield IDT kit. And these are for customers to express large-scale amounts of protein that are generally used for purification. Next is our one-step heavy IDT kits, which is basically our human coupled system that allows you to incorporate isotopic amino acids. The advantages of using our one-step IDT translation systems is you get higher protein yield. You have the ability to express large proteins that contain post-translational modifications. These proteins are directly compatible with colorimetric and fluorometric assays, and we have very high lot-to-lot reproducibility. What is needed to express in our system? First, you need your coding sequence of the protein you would like to express. Second, it is imperative that you have these DNA elements also in order to get the levels of protein that you need for your experimentation. Things that you will need include a T7 promoter element, an EMCV iris, a poly A tail, and a transcription terminator. How do you get these DNA elements into your, or together with your coding sequence? You can do this by multiple ways. First, you can use just general cloning methodology by moving your target into one of our many PT7 CFE expression vectors. Alternatively, you can see if your target or protein of interest is already cloned into an expression vector through a website at ASU where they have a DNA, it's called DNASU, and they have a repository of over 7,000 human clones that are ready to express in our system. Alternatively, one can use PCR to add these DNA elements onto the coding sequence of the protein that you want to be expressed. Lastly, we have recently engineered some PT7 CFE gateway-compatible destination vectors with various tags, and these are available upon request. So next, I'd like to go into detail information and data regarding our kits. I'd like to start with our one-step human coupled IDT kit. And again, this kit is used for small-scale expression. It comes in two different sizes, and the reaction sizes are 25 microliters, and the protein yield you get will be about 40 to 100 microgram per mil, with reaction time being protein dependent for 90 minutes to 6 hours. This is what your setup will look like. You will take your DNA or mRNA that contains the required DNA elements, and you will mix them with lysate and reaction mix into a tube, incubate at 30 degrees, and voila, you will have expression of your protein of interest. On the right, we have an example of GFP that is expressed in our human coupled system. And you can see for GFP, expression increases over time with about 100 micrograms per mil after 6 hours of expression. We are comparing this to a fluorescent GFP standard so that we can get the exact amount of GFP that is being expressed. Inset here is a picture of the GFP that is expressed after 6 hours when looked at under UV light. Next is an article that was published by Hartzau et al. In Biotechniques, where they were comparing relative expressions from 3 different IDT coupled systems. They were expressing 5 different proteins that all contained a CMYC tag, and here I'm showing you data from expression and then STS page Western blot analysis detection with a CMYC antibody. First, you see expression from our human IDT system, and you can see that we have strong expression of all 5 proteins that were expressed with very low background, compared to only 1 protein being expressed in the rabbit or tick system and a very high background, and undetectable levels in the wheat germ also with very high background. showing the superior performance of our one-step human coupled system. We have also seen this increase in expression when we compare expression from our coupled system with the Promega rabbit retic system. Here we're looking at non-radioactive labeling. We're incorporating Vo-Dippy conjugated lysine into IVT-expressed luciferase. On the left is an SDS page gel detecting Vo-Dippy conjugation, and you can see much higher levels of expression when luciferase is expressed in our thermoscientific one-step coupled system compared to the amounts of luciferase that are expressed from the Promega TNT system. Most exciting is that activity of luciferase that is expressed in our thermoscientific one-step coupled IVT system is much higher compared to the undetectable levels of activity that we see for the luciferase that is expressed in the Promega TNT system. This activity is further shown here when we express an active human caspase-3 antibody. Shown on the left is expression, where we are detecting with a specific active caspase-3 antibody, showing very high levels of expression. And furthermore, when we use a caspase-3 specific activity assay measured in relative luminescent units, we see very high levels of caspase-3 activity from the caspase-3 that is expressed in our human IVT coupled system. Next is a very interesting technology that was championed by Dr. Josh LeBaire, who is also giving a seminar in ExpressionWorld that you will be able to view. What this technology is called is NAPA, and it stands for Nucleic Acid Programmable Protein Arrays. And what it is instead of arraying proteins on a slide, we array full-length cDNA molecules on a glass slide, together with a capture antibody next to that full-length cDNA. We utilize our human-coupled IVT to transcribe and translate that protein, and once that protein is made, it is then captured by the capture antibody that is seated adjacent to that cDNA. This is used to query for protein-protein interactions. You can identify protein modifications and enzyme substrates. Here's an example of expression of proteins on NAPA arrays. Here we're looking at over 2,000 different GST-tagged cDNAs, and we're looking at the same cDNAs that are arrayed on 4 glass slides that were in vitro, that protein was in vitro translated with 3 different lots of our human coupled system on the left, compared to expression using the TNT rabbit retic system on the right. The proteins were detected using an anti-GST antibody and a CY3 secondary, such that red is denoting the highest levels of expression. And you can see you have much higher levels of expression in our system compared to the rabbit retic system. Furthermore, if you look to the right, the graph shows an excellent correlation between independent lots of our one-step human coupled kits. Next is a specific NAPA experiment in which they were immunoscreening NAPA protein arrays. This is data from Dr. Sean Rollins, and they studied cholera. In this case, we have the cholera orpheome on the array, and they were screened with sera from cholera patients that were either at day 0 or 18.5 months later after they were vaccinated with anthrax. Shown on the left are proteins that were transcribed and translated with the TNT rabbit retic system. Shown on the right are proteins that were transcribed and translated using our human coupled IVT system. There were no hits that were identified with the rabbit retic system. Because our system expresses so much higher, to a much higher level, they were able to identify multiple putative novel antigens. Another assay that has recently been developed that also takes advantage of the unique characteristics of our human-coupled IVT system is a high-throughput transcription factor assay developed by Dr. Mike McCoss at the University of Washington. Targeted proteomics is a powerful approach that enables quantitative analysis of tryptic peptides from complex biological samples. However, a major bottleneck of this limiting wider application has been identification of optimal proteotypic peptides that are readily detectable by mass spec. This is further complicated by low abundance proteins. To combat this, the McCoss lab in vitro synthesized over 800 different transcription factors to identify optimal proteotypic peptides and fragmentation patterns. They obtained these 800 transcription factor clones from the DNASU DNA repository. They in vitro transcribed and translated using our human-coupled IVT system, and then they purified these full-length proteins based on a GST tag that was at the C terminus. They digested proteins with trypsin and then monitored all tryptic peptides using SRM. They were able to identify optimal proteotypic peptides for each protein and fragmentation patterns for these peptides. Shown on the bottom right here is an example using a standard peptide, and here we see the fragmentation pattern and the subsequent fragmentation patterns from that peptide. On the top panel, we're just looking at expression, probing an SDS page gel with anti-GST of 4 different transcription factors that were then subsequently purified with glutathione, and here's a silver stain showing that purification. This is further shown in the following slide, where we have over 46 different transcription factors that were expressed, and we have expression shown on the top panel, again an SDS page gel detecting with an anti-GST antibody, and then the subsequent purification, silver stain, where we glutathione purify these transcription factors. One thing that was found in this study was that 2 glutathione proteins also co-purify that are endogenous to the system, and then they were able to be identified through mass spec and then taken out of the equation when they looked at their data. So they were able to detect proteotypic peptides, but were they peptides that would be useful in a lysate? And how to test this, I'm showing you an example of proteotypic peptides that were found for the transcription factor c-Jun. These are what the fragmentation patterns for that IVT expressed protein look like. And then they compared that to what the fragmentation patterns would look like for proteins in the nuclear extract. And what they found was that the relative contribution of each fragment ion to each peptide peak was similar, and that's displayed as different colors. And then just to show you with a different proteotypic peptide, you again have very similar distribution patterns of those fragmentation ions, showing that the proteins that were expressed in IVT are a good tool to telling you what proteotypic peptides you could use to quantify your amount of protein in a lysate. Next, I'd like to move on to talk about our one-step human high-yield IVT kit. And again, this kit is used to express higher amounts of protein. Here you will get a protein yield of about 250 to 750 micrograms per mil. Reaction time is a bit longer, 6 to 16 hours, and it comes in a couple different sizes, 100 microliter or 2 mil. We term these 2 different sizes our mini, which is shown at the top here, which is 100 microliters, or our maxi, which is a 2 mil reaction volume. And this is shown here as the workflow for how you would utilize our high-yield IVT kits. The mini kit comes, and again, this is a dialysis methodology, which enables us to remove inhibitors of translation as proteins are being expressed, and also add back ingredients that are being depleted as a protein is made, like energy, for example. So the mini kit comes as a dialysis unit that is inside a 2 mil tube. What you will do is remove that dialysis unit and set it aside on a clean part of your workbench, and then you will make up your 1x dialysis buffer and add that into your 2 mil tube. You will then add back in the dialysis unit into your 2 mil tube, and then you will set up your reaction, which at this time, instead of a 25 microliter reaction, it will be a 100 microliter reaction that will then be inserted into the dialysis unit through a port at the top. The 2 mil tube will then be closed. Alternatively, we have our maxi unit, which will express a 2 mil reaction volume, and you have a similar workflow. Again, everything is just larger. The 2 mil dialysis unit comes in a conical. You will then remove the dialysis unit, and it is large enough such that you can simply just invert it and set it upside down on a clean part of your workbench to make sure that you don't contaminate any of the dialysis membrane. You will then make up your 1x dialysis buffer, put it into your conical, insert the dialysis unit, and then put in your 2 mil reaction into that tube, further close the tube, and then both of these will then be incubated at 30 degrees for the protein-dependent amount of time required to express the protein of interest. Most customers who will be expressing a large amount of protein will need further purification to perform their downstream applications. For that, we provide different expression vectors for purification, where we have tags located on the C terminus and the N terminus of our P-T7-CFE expression vectors. Different tags that we have available include HIS, HA, CMYC, FLAG, and then multiple tags for the use of purification. You can subsequently do 2 purifications to get extremely clean protein at the end. Those contain GST, and for those, we also contain an HRV3C protease site that enables on-column cleavage of your protein to get pure protein purification. Shown here is an example of proteins that were expressed in our human high-yield IVT system, and then they were purified over immobilized glutathione. This is the PT7 CFB expression configuration. We had an N-terminal His followed by GST with an HRV3C cleavage site, the gene of interest, and then we have an HA located on the C-terminus to allow for full-length determination, full-length expression determination. From start to finish, you will have purified protein in less than 2 days. The workflow is as follows. You express your protein for about 7 hours. Start it in the morning of the first day. You will bind that protein to glutathione overnight at 4 degrees. Then you can either elute with glutathione to elute off the GST fusion protein, or you can on-column cleave to remove the GST tag and then elute your pure protein. Shown on the right is a Kamasi stain gel of 5 different proteins that we expressed and purified with this methodology, where we either added HRV3C or not. Shown here is GFP, GAPDH, green Rinoa, BAD, and C-FOS. And you can see very clean levels of purified protein, whether it's the GST fusion or the pure protein once you have cleaved off that GST tag. We also found for the protein BAD, shown here with this star, denotes a co-purified of a protein that normally interacts with BAD that I'll talk about later on. Here's a study by Saul et al. Who did a high throughput protein expression and purification comparison of our human high-yield system compared with the wheat germ and E. coli IVT system. They expressed 36 different proteins and looked at protein expression, which I'm showing on the left, and then protein purification, shown on the right. And what they found is that our system was better than the other 2 systems in both protein expression and protein purification. We've also found that protein activity is higher in our human IVT couple kit. Here we're comparing the expression of 2 different luciferase molecules, Rinoa on the left and Cypridina on the right. And interestingly enough, Cypridina requires over 13 to 15 different disulfide bonds to be formed in order for it to be active. Here we're comparing expression in our human IVT system compared to in vivo expression in E. coli. We further subdivided the luciferase proteins that were expressed in E. coli into total lysate and soluble fraction of that lysate. And you can see in both cases for Rinoa and for Cypridina, when we assay for activity based on an activity assay measured in relative luminescent units, we get much higher levels of activity in protein that is expressed in our human high-yield IVT system compared to what is expressed in E. coli for both Rinoa and Cypridina. Interestingly enough, even when levels of expression of soluble Rinoa are higher in E. coli when we assay them with an anti-His Western blot compared to expression in our human IVT system, levels of activity are much higher, showing the formation of a very active, readily formed luciferase protein. Next, I'd like to move on to talk about our one-step heavy protein IVT kit. And this system is identical in product structure to our one-step human coupled IVT kit, except for the addition of the heavy amino acids lysine and arginine. What it is used for is to identify ideal peptides for targeted quantification, similar to what Dr. Mokasa's lab was doing. It allows you to verify protein digestion in your mass flow, in your mass spec workflow, and you can generate controls for protein affinity enrichment loss. You can also use it as a quantification standard to measure changes in light samples. Shown here is the workflow. You would express your protein. In this case, you would express in a coupled system, purify, digest your proteins, run them through a mass spec, and then you would have your light and heavy versions of your protein. Just as with our human coupled IVT system, here's an example showing you a time course of GFP expression that is very similar to what we see in our coupled human system. You get about 100 micrograms per mil after 6 hours. More importantly, the percent incorporation of your heavy amino acids gets to be above 90% after 6 hours of reaction time. So next is an example of bad that we expressed in our heavy IDT system. And they found, shown highlighted, are the tryptic peptides that were identified, and that trace is shown here, where you can see the light on the left and the heavy peptides on the right, which we had greater than 95% isotope incorporation. Furthermore, BAD was shown to be specifically phosphorylated based on mass spec, shown here in the inset, alternatively also shown based on a Kamasi-stained gel looking at the purified BAD that also which I sort of talked about earlier, co-purified with 1433, where this interaction can only occur when BAD is phosphorylated. Next, I'd like to talk about our one-step CHO high-yield system, which is very similar to our human high-yield system, but because CHO is able to grow to a much higher density, it is a more cost-effective system for expression of large quantities of protein. Shown here, similar to our human high-yield IDT system, it is a protein purification workflow that, again, from start to finish, will give you purified proteins in less than 2 days. You express your proteins for 7 hours, bind to glutathione overnight, and in this case, we simply eluted with glutathione. So we're showing you a Kamasi-stained gel of GST-tagged proteins, 11 different proteins, and the same configuration as we showed with the human high-yield system, an N-terminal his followed by GST with an HRV3C cleavage site and the gene of interest followed by an HA. Again, you get Kamasi-stainable, purified proteins in less than 2 days. With the CHO high-yield IDT system, we have focused on the expression of active therapeutic proteins. Here's an example of expression of the human granulocyte colony stimulating factor, which is a 20 kilodalton protein that contains 2 intermolecular disulfide bonds that are necessary for GCSF bioactivity. Shown on the left, it is his tag, is a his Western blot showing expression of GST, and further on the right is purification through that his tag, showing you very clean purification of GCSF. This purified GCSF has been used to measure activity based on a GCSF potency assay measured in international units per mg, and you can see that the GCSF that is produced in our CHO high-yield IDT is active. So GCSF is expressed, purified, and active. Right now, you're probably asking yourself, well, how do I choose? Do I express my proteins in the human system, or do I express my proteins in the CHO system? What these data are showing is that relative levels of expressed and purified protein are similar between the HeLa and the CHO. So it'll be determined based on what is the common cell line used in your lab. Alternatively, if you need a more cost-effective, need a high level of protein, you could select CHO. Shown here are 3 different proteins that were expressed either in HeLa or CHO, and they were purified using glutathione, and they were either eluted on the left with glutathione or cleaved on column with HRV3C. Shown is the proteins RB1, streptokinase, or EPO, all that were able to be expressed and purified in both systems, HeLa and CHO, with comparable levels. Next, I'd like to show you some post-translational modifications that are added in our IDT system, both our HeLa and our CHO high-yield IDT systems. Here we have expressed P53 in a his configuration or GST, and we also expressed a non-related protein, CCND1, as a control. And then we ran these on STS page gels and detected with different antibodies shown on the right. In the first panel, we're just detecting with P53, showing you that you get expression of P53 in our HeLa and CHO in the his configuration and the GST configuration, HeLa and CHO. If you look down to the right is our control, where we're detecting based on the tags that are expressed with our proteins. You can see that you do see expression of our non-specific protein in that panel. So we are getting expression of that. Most excitingly, we detect with our post-translational specific antibodies. In this first panel, we're looking at a phosphospecific P53 antibody. We do see expression of phosphorylated P53. Furthermore, we see methylation and acetylation in both our HeLa system and our CHO system. When customers want to utilize our CHO high-yield system and they want to express proteins that will be used for therapeutics, one thing that might be important is to know the relative endotoxin levels. What we found is that when we looked at endotoxin levels in our IVT expressed proteins, they ranged from about 100 to 300 endotoxin units per mil. Just by simply purifying, we reduce those levels to the range of less than 0.1 to 15. But if you have a very stringent requirement that you need to be below 0.1, we suggest that you can simply pass your purified protein through our Pierce high-capacity endotoxin removal resin without any loss in protein, and then you will be at the undetectable or very low level of 0.1. Lastly, I'd like to talk about a new service that we have in development for customers to have their proteins expressed and purified, if needed. And what this service entails is that a customer will send their protein or DNA sequence to us. It will be gene optimized and synthesized into string DNA moieties. These will then be stitched together with the elements that are required for our IVT system, and then the protein will be expressed in our IVT system and purified if needed. Highlights of the service include that the genes are optimized and synthesized, and that gene synthesis to supplying the purified expressed protein is about 10 days. To show you the robustness of this system, over 100 different string moieties were expressed into 100 different proteins, and we took those proteins from a number of different types of proteins, and here they were all His tagged. So I'm showing you a Western blot using an anti-His antibody, showing you the expression of those antibodies, in which we had a success rate of over 95% for protein expression. In summary, our one-step in vitro protein expression kits provide rapid production of functional proteins. Our one-step human and CHO system is capable of expressing over 250 microgram per mil of protein. The protein expressed in our one-step human high-yield IVT kit has a higher activity than the same protein that was expressed in E. coli. And proteins that are expressed in our kits have relevant post-translational modifications and can form protein interactions, as shown by protein 3,3 and NAT. I want to thank all the people that helped make this kit as great as it is.
That was a wonderful presentation. Thank you, Dr. Jensen. Next, I'd like to introduce our next speaker, Dr. Govind Rao. Dr. Rao is Professor of Chemical and Biochemical Engineering at the University of Maryland, Baltimore County. He obtained his PhD from Drexel University in Chemical Engineering in 1987. Dr. Rao's research is targeted towards application of fluorescence spectroscopy to create novel low-cost sensors for bioprocess, biomedical, and environmental applications. His lab has developed next-generation sensors for low-cost, non-invasive monitoring of oxygen, pH, and PCO2 in bioreactors. He has been a faculty member at UMBC since 1987, and in 2006 he founded the Center for Advanced Sensor Technology, or CAST, and has been serving as its director since. I will now turn it over to Dr. Rao for his presentation.
Hello there. So following up on Dr. Jensen's presentation, I'd like to set the stage for how extraordinarily useful the thermo IVT system has proven to be. In 2012, DARPA held a public information meeting where they described the need to have the ability to manufacture a variety of drugs at the front line. And this was really driven by the need to have the ability to have increased medical capability for far forward providers with recent deployments that have been under very harsh conditions. It was clear that in order to serve the people on the front line, you need to have the capability to produce various pharmaceuticals on demand. And the idea was that you could then get rid of the entire supply chain that would allow you to have a system that you could flip a switch and produce whatever medication you needed without carrying a large inventory, or in the case of biologics, a cold chain. So the initial program looked at small molecules that would be in a tablet formulation, and that was then extended to a biologically derived medicines on demand concept. And at this information session that DARPA held, they laid out some guidelines. They wanted to see a system that could be very generic, that would allow you to have a great deal of flexibility to produce a variety of sequences to basically encompass an entire range of protein-based therapeutics. It had to be very flexible and portable that would allow you to make whatever you needed within 24 hours. And the other thing was that it had to have a complete end-to-end manufacturing chain that included purification. And they set the bar really high by requiring that these biologics or protein therapeutics that were produced would have to be something that were already FDA approved. So in other words, a biosimilar. And so this meant that it would have to be a molecule that would have the kind of quality, consistency, potency that you might expect from any injectable therapeutic that's available today in hospitals and clinics across the developed world. And so they left the platform fairly agnostic in terms of whatever technologies that needed to be developed. It could be cell-based or cell-free. However, when I attended this meeting, I could not conceive of a system that was cell-based that could work and produce therapeutic protein in less than 24 hours, because it just takes cells at least that amount of time to even divide. This is even with the fastest growing E. coli. To grow to a density where you could harvest reasonable dosage would require several hours, and then let alone the recovery and purification, which would need another several hours. So 24 hours just seemed wildly unrealistic. And as far as the mammalian system goes, with a doubling time of 20 hours on average, there was no way to meet this target. And that was bad enough, but then the rest of the challenge was also to develop an integrated platform that would have everything on board. About that time, I came across an advertisement from Thermo, which talked about their wonderful IVT system. And so I approached Thermo to be a part of a consortium that would then go and propose a solution to DARPA that would use the cell-free system as the heart of the expression platform. And Thermo readily agreed to be part of this. And so this slide just lists the different consortium members who are part of the DARPA program. And we have successfully gone through phase 1 and are now in phase 2. And our goal is no less than a re-engineering of the entire pharmaceutical industry model. So this slide here shows you a conventional manufacturing facility that's cell culture based, that produces a protein-based therapeutic, and was from a slide that Dr. John Birch at Lonza had used to describe in a paper. And as you can see, there's a very complex train of operations. You start from a master cell bank where your genetically modified cells are kept in liquid nitrogen. You gradually scale the master cell bank up through a series of expansions in volume, and then go into a production train that might require reactors on the order of several hundred or several thousand liters. And then you have to, depending on the product, if it's within the cell, as is the case for most E. coli-based systems, or a secreted product, which is the case for most mammalian systems, depending on the particular product, go through a series of recovery steps, which then allow you to purify the product of interest, and then eventually stabilize it, formulate it, and ship it to your point of care. So a good analogy here would be to imagine if you had to depend on all your food coming from a distant plant and shipped to you either in cans or frozen. And so what we're trying to do is upend this paradigm to some degree and return the process to the point of care with a compact modular system that would be able to produce your therapeutic at the point of care. So to use that food distribution analogy, we're now creating the ability to give each facility its own kitchen where they can make freshly prepared food. And that's a pretty useful analogy because it allows you to then immediately grasp the significance of what we're trying to do. You do away with the entire current centralized manufacturing paradigm, which is kind of a throwback to how the industrial age developed, and really empower caregivers with the ability to produce biologics of various kinds at the point of care. So imagine now at someday if you could make your own insulin or make your own antibody for administration either in the doctor's office or right in your own home. That's sort of the paradigm that we hope this technology will lead to. And Thermo did another amazing thing when they figured out that you could actually not just work with the cell extract, which is amazing enough, but also you could lyophilize it. So what that does is eliminate the entire cold chain. And once you've eliminated your cold chain, you now have the ability to really stockpile your raw material in a lyophilized formulation. But how well does this work? So we did some test studies, and you see a vial on the left, which is the lyophilized formulation. And the students who got this were very excited. So what they did was they took 3 vials. The middle one is a control, and the one that's labeled incubated was actually put in a controlled temperature environment with a GFP plasmid added to it. And the field-tested one was simply one where the student reconstituted it, added the GF plasmid, and then just wandered around with the vial in their pocket to simulate someone who might not have a laboratory with an incubator. And amazingly, both the incubated and the field-tested formulations glow nice and green, whereas the control shows no expression, as you might expect, showing the great versatility that this platform has from meeting not just DARPA's needs of use in battlefield medicine, but potentially even in a whole host of other applications. And some of their data here just showing the early studies that can make FDA-approved therapeutics like erythropoietin at single band purity in less than 2 days. We've since advanced this quite a bit more, and we're well on our way to meeting the under 24-hour target. In another case, streptokinase was also a simple module system, and all these are produced with very high potency and activity. So we have a lot of confidence that eventually this could lead to a shift in the current manufacturing paradigm. The other thing that we're trying to do is build in a level of rigor into the manufacturing process, because you're now going into a new realm, because you're trying to produce a biologic at single doses. So how do you actually ensure that you have a very consistent product? And the only way to do that is to make the manufacturing conditions so precise that it's no different than how you would make an iPhone or a laptop computer. The idea being that if you build in that rigor into the production process, then at the end of it, every single dose that's produced will behave the same way. And so we set out by using many bioreactors that were highly instrumented, and this is just a picture showing some of the early prototypes. But more importantly, if you look at the kind of production profile you get, note the remarkable consistency of expression in 3 separate bioreactor experiments. They all behave exactly the same way. And the other remarkable thing to note is the process time. You're done in 2 hours from start to finish. You have expressed pure protein very, very quickly, leaving you the rest of the time to purify the product and get it into whatever format you need for delivery to the patient. So this, we believe, will spark a new revolution in how therapies are developed. And here again is data from the same 3 bioreactors showing you how remarkably consistent the recovered protein is. This is just UV irradiation of GFP showing that they all glow nice and green uniformly. The other side is purification, and here we're working with David Wood's lab at Ohio State, who is developing a generic purification strategy based on inteins. Inteins are these self-cleaving chains, polypeptides, that based on either a pH switch or a temperature switch are able to self-cleave, and thereby it allows you to generate pure protein of your product, shown here in blue. So the binding domain is shown in green, and the intine is the sequence in orange. And so once you express it in the IVT system and you bind it to your column and you switch the pH and elute it, you essentially can recover your pure protein, because of that low pH buffer, the intine starts self-cleaving. So this is again something that has been shown to work very well. Here is the proof of the pudding with streptokinase. The cleavage was induced, and you can see that you're getting essentially a single band of streptokinase at the end of the process. So we're now working on a variety of different proteins with the same technology. And the advantage here is that you're able to make a product that is completely tagless. And so there is no expression tag in your final formulation, which is what would be desired in a therapeutic that you're going to inject into someone. Now, this is clearly the work of an entire team, and this is just showing you a slide of the entire team that's behind this project. And looking at what's on the center table is a prototype of this suitcase-sized device that can produce and purify the protein. And it's been an amazing journey that's still continuing. We're just 3 years into the project and are continuing to push forward. And I'd like to end with disclaimers that this is not endorsed either by Thermo or by DARPA. And I thank you very much for your attention.
All right, that was an excellent presentation, Dr. Rao. I'd like to thank both Dr. Jensen and Dr. Rao for bringing the information to us. And before we get started on all of your questions, here's a quick reminder about how to reach us today. Questions can be sent via the green Q&A button at the lower left of your presentation window. We'll try to get to many questions as we can today. So our first question today, a participant asks, what is the main differences between your system and other available IVT systems?
The main difference is high protein yield with the addition of post-translational modifications. And again, we have both the human system, HeLa, and the chill-based system.
Great. Next, one participant asks, what is the largest protein expressed in your system?
The largest protein we've been able to express is mTOR, which is greater than 250 kilodaltons.
Interesting. Next, one participant asks, has there been any proteins that have not been able to be expressed in your system?
In terms of expression, we have not had a problem expressing proteins at this time in either the human HeLa-based system or the chill-based system.
Great. We have time for one last question, and if we didn't get to yours, we'll follow up with an email. And in the last question, a participant asks, can multiple proteins be expressed in your system?
Yes, you can simply add multiple cDNAs with the required DNA elements, and expression can be of multiple proteins. We do find that we get a slightly lower level of yield of protein because you're expressing the different proteins. But yes, we would express up to 5 different proteins in our IVT system.
All right, fantastic. I would like to once again thank Dr. Jensen and Dr. Rao for their presentation, and thank you to Gibco, part of Thermo Fisher Scientific, for making today's educational webcast possible. Today's webcast will be available for on-demand viewing through May of 2017. You will receive an email from us alerting when it's available for on-demand and posted on labroots.com. You're welcome to forward this to any of your colleagues who weren't able to join in today. Thanks for logging in, and we hope to see you again soon. Bye-bye.